In this video, we will learn about a technique called the Punnett square. Now, this technique was developed by a British geneticist by the name Reginald C. Punnett. And this geneticist is best known for developing this graphical method or a diagrammatic method to determine the genotype of organisms. Now, let us elucidate it by taking an example. For example, a plant breeder, or this is true for even animal breeders or geneticists. Let us just take an example. For plant breeder, let us assume a plant breeder wishes to know what will be the outcome of a certain cross. What will be the phenotype and the genotype of the offspring if he crosses two plants? Now, let us assume he has got two plants, both of them are tall, and he knows, and he should know basically, he knows that this is the characteristic, both of the plants are tall, and the genotype of the two plants are also known. For example, let's say the first plant here, which we are considering as a male, by which we mean this will be used as the source of pollens. Now, this plant here is, let us say, heterozygous for this particular trait. What do we mean by that? Is it has in its allelic pair, and here is the allelic pair. In the allelic pair, each allelic partner basically expresses a contrasting trait. Similarly, the other plant is also tall, but this is homozygous. So you see the phenotypic expression, which means both the plants are tall, but genotypically they are different. The first plant, which is supposedly the male now, which we are considering to be a male, is heterozygous, but the second plant is homozygous for tallness, by which we mean that each allelic partner is identical. Each allelic partner is identical in the allelic pair. This person Punnett, he devised a method. So he basically made this particular graphical diagram in which he used this to figure out the combinations of the different alleles to determine the outcome. Now let us see. Let us just illustrate it how it is basically done to understand it clearly. So let us look at our first plant, which is a heterozygous male. Now it can be a female, it doesn't matter, but as convention, the data for the male is written onto the top, and the data for the female is written to the side of the Punnett square. Now let me just rub it, make it a little bit clean so that it doesn't become way too messy. Now let us look at our heterozygous pollen donor. During the process of gametogenesis, when the gametes are being formed, these two allelic partners will segregate, right? And 50% of the gametes that are formed will receive the dominating allelic partner, and 50% of the gametes will receive the recessive allelic partner. Now let us look at the homozygous plant. Now in this homozygous recipient, or the female plant that we are considering a female plant, the two allelic partners will also separate, and half of the gametes will receive 50%, half of the gametes will receive one of the allelic partner, and the other half will receive the other allelic partner. But basically both, because it's a homozygous plant, or this condition is homozygous for the trait of tallness, it basically just doesn't matter, because all the gametes will have capital T, or the dominating allelic partner. Now, what is done in a Punnett square is that the genotype of the gamete is written. So let us write the genotype for the organism. So there is only a 50% chance. So we only need basically two squares for this. So we will write capital T here. Let's write capital T, which are the gametes formed by the male. So we write T here, which comes from here, and we write small t here, right? So we write small t here. Okay, and let me rub. I hope you've understood this part. So let me just rub up the mess. Where is what gamete coming from? And from the homozygous female parent, the genotype of the gamete is T and T. So half of the gametes have the genotype T, and the other half also has T. So these are basically the gametes, the genotype of the gametes that are written on the top and onto the sides. Now let us start developing the combination. So what is the combination when these two gametes, let's say this particular pollen fertilizes this particular ovum inside the plant body, then what is the combination? So we get a capital T and a capital T. And the other one, we get a combination of capital T, small t, then we get the capital T, and we get a capital T and a small t. Right, so these are the four possible outcomes. These are four possible outcomes. It is not necessary that these four will be generated, but these are the four possible outcomes if these gametes fertilize, right? So these gametes come together and enter a fertilization event. Now, here we've just picked up only one particular trait for analysis, which is basically the trait of height. But a Punnett square can be drawn for more than one allelic pairs, by which I mean you can basically make one Punnett square to study, let's say, two traits or three traits or four traits. So when we are only developing a Punnett square or conducting an experiment keeping only one characteristic in our mind, we call such an experiment or a cross as a hybrid cross. So hybrid cross. And if we are conducting an experiment taking two characteristics into an account, we call it a dihybrid cross. If we are taking into account three, we call it a trihybrid cross. If we are taking four into account, we call it a tetrahybrid cross, and so on and so forth. But as the number of characteristics for study increase, the complexity of Punnett square also increase, which we will study sometime in the future. Now, what is the outcome of this? Now, all right, we have made the combinations. We can now see the possible combinations, but what is the use of these particular, knowing these four combinations to a geneticist or a plant breeder or an animal breeder? Well, the outcome is this. The geneticist, a plant breeder, animal breeder, or anybody interested in knowing the in basically conducting the Punnett square test can determine two things. This person can determine the phenotypic ratio of the offsprings and also the genotypic ratios of the offspring and the phenotypic ratio of the offsprings. Now let us find out were we able to do that through this Punnett square. So here we see there are how many genotypes are possible. So there are two genotypes possible, and the first genotype, and let me use a different marker, let's use a red one. So you see the first genotype that we get is capital T, which is homozygous dominating. So let's write the first genotype here. So it's TT. And how many such genotypes do we have here in the Punnett square? So we got one and we got one here. So basically we got two genotypes here for homozygous dominant. And what are the other? All right, again we have capital T and small t, which is a different genotype, capital T and small t. And how many do we have? We have got like two combinations here, two possible combinations, two possible outcomes. So let's also write it T and T. So what is the ratio? The ratio is 2tt is to 2 capital T small t. And if we simplify it, the ratio becomes basically 1:1. So 1:1. So what we can conclude from this is that the outcome of a breeding experiment between a plant which is heterozygous for tallness and homozygous for tallness will, or rather has the probable outcome of giving us 50% plants with a homozygous dominating genotype and 50% chances of having a heterozygous condition for the particular characteristic. So you can determine the genotypic ratio, how many organisms will have a particular kind of a genotype. So this is called a genotypic ratio. Also, the plant breeder can determine how many plants will have a certain phenotype. So how many phenotypes do we have here? We have got like we only have one observable phenotype, which is tallness. Also, we have the recessive trait, which is not expressed here. So how many phenotypes are possible here? So what are the different phenotypes? So we got tallness and we got dwarf, right? So we got tall plants is one of the characteristics. We got dwarf plants. So how many tall plants do we have? Well, all of the four combinations that we get here are for tallness, right? because they have a dominating allelic partner in them at least. And how many dwarf plants? We get zero. So what is the phenotypic ratio? The phenotypic ratio is simply 4 tall, yeah, or all tall plants. All the plants will be of the given phenotype, which is tall. I hope you've understood what a Punnett square is and what are the two major uses of a Punnett square. We will keep coming across Punnett square in our future classes when we study the laws of inheritance in greater details. Thank you.